Hey guys, this is Kendall Terry, and this is meiosis, the making of sex cells in essence, but it is how sexual organisms reproduce is really the start of this focus. So we're going to take a little journey here to discuss meiosis. So the start would be looking at family. Now this here is my family, of course, and you can see my kids here and my wife, and when you look at our kids, they do have a resemblance of us, but they don't look exactly like us. And that's all because of meiosis. And how those sex cells come together produces a unique individual. And notice when you look at my kids, although they show resemblance as well because they come from the same two parents, there is a difference in each of them as well. And that difference is because the chance of you getting two identical set of genes to line up together is pretty much impossible by chance. Now you see it in identical twins whenever you have one egg that's been fertilized by one sperm that is split into two individuals, but when you're looking at just on a child-by-child basis, there's going to be differences, and that's really what we're talking about, is how do those differences occur. So the first thing we need to understand then is, once again, that difference between haploid and diploid. We went over this once already with mitosis, but it's worth revisiting. So here we've got haploid and diploid. Haploid is just one set of the chromosomes, whereas diploid is two sets of the chromosomes. So the majority of cells inside of any of your body cells are diploid. They have both a set of chromosomes from your mom and a set of chromosomes from your dad. But whenever you're making sex cells, the egg and the sperm, then you only end up with one set of those chromosomes, and those cells are considered haploid cells. So to understand this, let's look a little bit at what we call the human life cycle. So here we have dad and here's mom. They each produce through meiosis, which is the process that we're talking about today, the sperm and egg, which are haploid cells. Those two haploid cells come together at fertilization, and they produce that embryo, which is a diploid or zygote, that diploid fertilized egg. That will, through mitosis, that we've already discussed, produce the child, produce the adolescent, produce the adult, and then once again through meiosis, you will produce those sex cells. So what do they look like? What do those chromosomes look like whenever we're talking about those today? What we'll refer to as homologous chromosomes. So you do get one set from dad, one set from mom. You can see them lined up here. Of course, the dad one's blue, the mom one's red. So these are chromosomes that are similar, the definition of homologous chromosomes, similar in size, shape, and genetic content, but they're not identical. So when you line them up, for instance, in the human, this would be a boy, and we know this because he's got the Y down here. But when you line these up, you see that there is a chromosome from mom, chromosome from dad. They're both similar in their size. They're similar in their banding patterns, which we talked about. That was just like looking at their genes. And whenever more research is done, they're similar in their genetic content, meaning that they are similar in actually what's going on there. So you've got, here's the two, one from mom, one from dad. Number three, one from mom, one from dad. So on and so forth, all the way down until you get to the sex chromosomes, the X and the Y. In this case, since it's a boy, you've got two that don't have a similarity. Now the reason is because the X chromosome is different than the Y chromosome. But when you look at a female, if a female was here, you would have an X chromosome from mom and an X chromosome from dad, and they would look more closely related. So what happens in meiosis? How do we use these homologous chromosomes? What does it all mean? Well, meiosis is split into two phases. Now, when we did mitosis, there was just one phase: prophase, metaphase, anaphase, telophase, and cytokinesis. But when we look at meiosis, there's actually two divisions that have to take place. So in mitosis, we ended up with two daughter cells that were genetically identical. In meiosis, once it's done, we'll end up with four cells that are genetically different. And let's look at that process a little bit closer. So the first one is meiosis I. And in meiosis I, of course, you know this because it's also got a 1 behind each of the names as well involved in the phases. Notice that the phases are still the same titles as mitosis, but there are some differences that take place. So in prophase 1, you get the nuclear envelope dissolves just like in prophase of mitosis. But here, instead of just those X's being visible, those chromosomes being visible or folded up, the homologous chromosomes will actually line up next to each other, and you will have a twisting go on of these chromosomes. So you've got two X's, one from mom, one from dad, and when they come together, they start to twist around and wrap around each other like this. Now, the importance there is that it's going to create a process that we'll discuss later called crossing over. So when we discuss that later, if you need to, you can go back to look at this right here. But this is what we're talking about, and you can see the pointer on the screen, of crossing over occurring in prophase 1 of meiosis. Of course, the centrioles go to the poles and the spindle fibers appear. Then in metaphase 1, just as in metaphase of mitosis, the chromosomes line up in the middle or on the metaphase plate, if you will, of the cell. Now the difference here, and you can think about the difference, you can go back to your notes and look at it, is that instead of the chromosomes just lining up one on top of the other, they line up beside each other. Now part of that reason is because they're all twisted together like this. Every chromosome, every homologous chromosome is all twisted up and folded like this, so there's no way for them to line up on top of each other. They're connected right now. So they line up in the middle beside their homologous chromosome. And you can see that here, the red and the blue one, and the homologous chromosomes, the ones that are similar in size and shape and genetic content, all right there together in the middle of the cell. Then you have anaphase 1. Anaphase, those spindle fibers start pulling at those centromeres, and it will pull these apart, the homologous chromosomes. Notice, though, that they're still connected at the centromere to their sister chromatids. The sister chromatids are still connected. In mitosis, you have them separate in anaphase. That doesn't happen here yet. So the sister chromatids that were beside each other, folded up, will separate. Now, the thing that you want to notice here is when that separation takes place, you actually have an exchange of DNA. So now you've got, this is an X, a red X, so that one came from mom. It's got a little bit of blue here on the end of it because there's been an exchange of DNA. Whenever that crossing over was all wrapped up, some of dad's broke off and exchanged with some of mom's chromosomes. So you get a little difference in those chromosomes now than what they were at the beginning. Okay? And then telophase 1, the nucleus reappears, and you end up with two cells. Now, if you go back and look at this cell here, this was a diploid cell. And if you count the chromosomes, 1, 2, 3, 4, 5, 6 chromosomes in that cell. When you go over here and you look at the number of chromosomes here, technically you have 1, 2, 3 chromosomes and 1, 2, 3 chromosomes. You're reducing the chromosome number in half. Now the problem here is that the centromeres here are still connected to sister chromatids. If those would separate, technically each individual one there, because those are copies of each other, would be a chromosome. So although technically we have 3 chromosomes, if that centromere separates, you could say that that cell has 6 chromosomes. So the number really, although it has reduced, hasn't gotten to the point that we need it to get to yet to make those sex cells. So there's another phase that has to take place. So here is, this is meiosis II. So this is the end of meiosis I right here, telophase 1 and cytokinesis. Notice that there's 2 cells. That's why you've got 2 lines here. So you've got 2 cells that are each having to go through this process. So we're only going to look at the top part. It's the same thing happening on the bottom. So prophase 2, the nuclear envelope dissolves, centrioles appear, spindle fibers grow. You can, of course, see those chromosomes. Metaphase 2, they line up on the plate. Now notice here they line up right on top of each other. Okay? So this meiosis II is more like what we look at in mitosis. Okay? They line up in the middle. Anaphase, that centromere splits and the chromosomes go to opposite sides. And then telophase 2 and cytokinesis, the nucleus reappears and those cells will split in 2. So now you end up with 4 cells. But look at the number of chromosomes. Remember, our diploid number that we started with was 6, right? We had 6 chromosomes. Let's count them now. 1, 2, 3 chromosomes. So we've reduced the number of chromosomes in each cell by half. So we've created haploid cells, or just N. In algebra, we would designate it as just an N. Diploid is 2N. Okay? So in humans, the haploid cell is 23. In each organism, there's a different number. So you can look those up if you're interested in how many chromosomes a particular organism has. You can look that up. In humans, our diploid number is 46. That's all of our body cells that you see. Our haploid cells would be the sperm and the egg cell, and those are 23 chromosomes. So that whenever that sperm and egg come together, 23 plus 23 equals that 46 total chromosomes that's in each of your cells, except for those sex cells. So how do we get to, go back to that first picture, if you think about that I showed you of my family, there's still a difference that occurs. Well, there's several reasons for that. One of those reasons is because here, if you look at these cells, each of these are different. They're genetically different from each other. None of these are the same. In mitosis, you end up with 2 genetically identical cells. In meiosis, you end up with 4 genetically different cells. Okay? Now, there is a difference here between boys and girls. Boys, you will end up with 4 genetically different sperm. In girls, you only make 1 egg in this process, and then there's 3 what we call polar bodies that don't form anything. But each of those technically are a cell on their own, and they are different from each other genetically. But let's look now at this genetic variation that occurs. There's 3 ways that we say genetic variation occurs through meiosis that gives us all the variety, and why you probably don't look identical to your sibling, although you have similarities because you have the same 2 parents possibly. You're not going to have you're not going to be identical. The first is independent assortment. When we look at independent assortment, this is this happens in metaphase 1 of meiosis. And what happens is when these chromosomes, remember, they're all twisted together. I didn't show you all twisted together here because I wanted you to see how they line up. They're all twisted together and they line up. Independent assortment says that they don't have to line up the exact same from one homologous set to the next. In other words, this homologous chromosome is moms and dads. The second one here is moms and dads. When you look at this way that they could line up, you get mom, dad, and then down here you get dad and mom. So that creates a different cell. So in this cell you've got brown eyes, black hair. In this one you've got brown eyes, red hair. So there's a difference that's created. That's called independent assortment. Those homologous chromosomes don't have to line up with all mom on one side, dad on the other. They can line up in whatever order they happen to line up in. Next is crossing over. Remember, we said crossing over occurs in prophase 1 of meiosis I. And here's what that looks like. I was trying to show you with my fingers, but here you can see it in a picture. So here you've got, this is 2 homologous chromosomes beside each other. Here's crossing over. Notice all this twisting and turning back and forth. And then you get over here and you've got all this exchange of genetic material that has happened in these chromosomes. So you end up with chromosomes that none of these are identical to the individuals that started them. They all are different now. And this creates variation in the population. And variation is key to life. Last is fertilization. Out of the millions of sperm that are released, only one of them is going to fertilize that egg. And that is completely by chance. So you get a complete chance of which one's going to win the race, so to speak, and be able to fertilize that egg. So let's sum everything up from meiosis here. So meiosis reduces the amount of chromosomes in half. In humans, that's 46 to 23. And then genetic variation occurs, causing those sex cells to be different from each other and different from the parent as a whole as well. So you end up with offspring that are genetically different. You end up with variation, and variation in population is good. And those 3 ways, once again, independent assortment, crossing over, and random fertilization.